This is an amazing question, and the answers have just incredible implications. So we have here this enormous single-celled organism where this, particular size of specimen probably has upwards of millions of nuclei within one single cell. What does that mean for the genetics of this creature? What are the heritable traits? Can we have a specimen that has multiple different genomes within one single cell because it has multiple different nuclei? The answer is a resounding yes, and that happens more often than it doesn't, actually. Most of the specimens that you come across in nature are not pure specimens that have this one, contiguous copy of their genome. They are this mosaic of all of these different specimens from that region that have gone through several, if you will, generations of being separated and fusing back together, reuniting, and sexual reproduction. They have a stage for that as well. It's just an absolute mess of genetics, actually. In fact, we have in my lab, well, it depends on how you define it, but we have over a dozen natural strains from across the world. This organism lives on every continent except for Antarctica, and every region tends to have its own sort of average specimen, right? We have strains from Japan, which was the one that was used to solve the maze in that famous experiment. We have several strains from the US, one from North Carolina, several from the UK, and they all have slightly different just quirks about them. The one from Japan is a little more heat sensitive. The one from Carolina is actually pretty well rounded, but it's famous for its spore viability, actually. And one of them from the UK is called Daywalker because it is substantially less sensitive to sunlight and just ambient light than all of the other strains. It's like half blind, if you will. It doesn't have the same reactions to light as some of the other strains do. So they all have these different quirks, and most of them are cross-compatible. If you fuse them together, if you put them in the same container, they will still do exactly the same thing that all Physarum polycephalum specimens do, and they will locate each other with the scent and fuse together and just run towards each other as quickly as possible to fuse into the largest cell that they can. And then, of course, you end up with this fusion specimen with all of these different traits that intermingle in just strange ways. And it's absolutely fascinating, but also kind of frustrating in some cases. Like in my lab, we spent a very long time trying to figure out how to get one of these genomes in isolation, genetically modify it, actually, in several helpful ways to study it and to have it do helpful things. That was an incredible challenge. I'll show you. We made this one, which expresses a green fluorescent protein, but only about maybe 1 to 3% of the nuclei in this particular specimen are expressing that gene, and the rest of them are just natural, just the native genome of whatever whatever mixture it was to begin with. And there are ways that you can purify this, purify and get it into single genomes, but it's very difficult, actually. And that's one of the challenges to genetically modifying this creature, is getting it down to one nucleus to modify, or selecting the one nucleus from this whole bag of them. And no, there is effectively zero segregation between these nuclei. They themselves, I don't think, have that much biochemical awareness, if you will, about the distinctions between them. I don't think that they could segregate themselves if they needed to. Anyway, so this leads to a bunch of very strange downstream phenomena where if you do have two or more strains that come together, some of their strengths and weaknesses can amplify each other or reduce each other, and you get all of these just hybrid strains, or they're not strains, but hybrid mixes of hybrid cells, where they have all of these different attributes, and they can help each other overcome some of their weaknesses. And it's very interesting, and you can see just how this incredible diversity could guide this creature through all these different scenarios and environments, and how this is actually a really interesting and beneficial trait. Oh, just fascinating. Great question. It's so strange.